to remove actually amine group of amino acids. Removal of the nitrogen from amino acid can occur by two different ways. The common way is the transamination. What does it mean when we say transamination? Transamination means actually transformation of amine group. If you transfer amine group from one substance and actually convert this amine to the another amine containing group known as a transamination. It means that the amine group is not released as a free ammonia. Okay? This is, for example, X amino acid contain ammonia. In the transamination, okay, this amino acid donates its amine to the, for example, another compound in a simple actually way. I want to show in a simple way the difference between transamination. We have a transferation of amine group from one compound to the another compound. As you can see here, the ammonia is not free. Okay, but when we say deamination, in a simple way actually. When we say deamination, it means that you remove the amine group from one substrate and liberate as a free ammonia. Okay?
Yes.
This is a transamination and this is a deamination. During the actually amino acid process, we have two different ways to removal of the nitrogen atom. And one of them is the transamination. For example, you want to start the actually catabolism of this amino acid. All the time, you require one keto acid, okay, as the acceptor of this actually amine group. Okay, for example, this is the amino acid and this is the alpha ketoglutarate. Alpha ketoglutarate accepts the amine group. Okay. So most of the time, alpha ketoglutarate regarded as the most acceptor of the amine group. After this conversion, you have the alpha keto acid and it is a carbon skeleton. Alpha keto acid is a carbon skeleton, okay? For example, this is a general structure. This is the general structure of amino acid. After removal of the amine or nitrogen by the maybe transamination or deamination, the remaining structure is carbon skeleton. Carbon skeleton known as the alpha keto acid. Okay, this is the alpha and this is the acid. Okay, so the remaining structure known as the alpha keto acid. And again, our body produces glutamate. When alpha ketoglutarate receive the amine from amino acid, they convert to the glutamate. This reaction known as the amino transamination. And we have some specific enzyme named as a transaminase. And this glutamate maybe participate and act as an amino group donor in the synthesis of non-essential amino acid, and sometimes it became dehydrogenated. So if I ask you, what is the fate of glutamate after transamination, you should mention that sometimes it's donated, because this is, for example, this is actually alpha ketoglutarate, okay? When alpha ketoglutarate receive the amine, now it is a glutamate, okay? And this glutamate sometimes actually can participate in a biosynthesis of amino acid. Okay, this is a glutamate, donates its amine group to the non-essential amino acid. Okay, but sometimes it became again dehydrogenated by the specific pathway, and I will describe in the next slide. And another way of removal of nitrogen we have a deamination, maybe occur in oxidative form or non-oxidative. For example, for non-oxidative, we have a serine and threonine. You know, we have a 20 amino acid that's naturally occurring in the protein, okay? The first step of the catabolism is the removal of the nitrogen, okay? It depends on the kind of amino acid, okay? They undergo to different reactions. Some of them undergo a transamination, okay, according to the type of amino acid, and some of them actually participate in deamination. For example, serine and threonine became deamination from the non-oxidative way by the specific enzyme, and the name of enzyme, for example, is serine dehydratase, threonine dehydratase. For example, alanine and aspartate became transaminated. So, you know, it depends on the nature of your amino acid. Some amino acid, according to their nature, became transaminated. Some of them, like for example, serine and threonine, they release their amine in a deamination. Some of them is oxidative, another one is actually non-oxidative. Okay, transamination, at first I want to start with the transamination. As I mentioned, the first step, and actually the obligatory step in the catabolism of amino acid, is the transfer of amino group to the alpha-ketoglutarate. And the name of enzyme is a transaminase or amino transfer. Both of them are similar. And if you remember, they produce the alpha-keto acids. The prosthetic group, you know, all the transaminase enzyme, maybe it is alanine aminotransferase, aspartate aminotransferase. All kind of aminotransferase require a B6 vitamin as a cofactor. Okay, B6 vitamin, if you remember the vitamin station and the function of the B6 pyridoxal phosphate, PLP. Pyridoxal phosphate is the active form of B6 vitamin, and one of the reactions that actually this vitamin act and function as a coenzyme is a transamination. So all kind of transamination required to the B6 vitamin. So in a deficiency of this vitamin, the digestion of protein would be defective. Okay? Because if you want to digest and start the catabolism of the amino acid, at first you want to remove the amine. You require transaminase, because these transaminases require B6. At the deficiency or situation of the B6, this reaction cannot occur. So the actually digestion of the protein is defective. All amino acids can actually undergo the transamination except threonine and lysine, and some references refer to the serine. Okay, some references mention that only lysine and serine, and some references mention that 3 amino acids cannot undergo the transamination. And are differ, for example, for L-amino acid, and the name is for amino acid donor. What does it mean when we say alanine aminotransferase, ALT and AST? This is a reaction of ALT and AST. These two reactions is very, very important. Okay. For example, if you want to evaluate the function of the liver, the first step, you know, we have some routine checkup, okay? For example, you want to check your FBS, triglyceride, cholesterol, okay? Lipid profile, and for example, glucose concentration. And also, as a routine test, most of the physicians order the activity of ALT and AST. These are the most important aminotransferase that can found in a high concentration in the liver. So generally, they consider as a liver function test. Okay? So the first step for evaluating the liver situation, you should order the concentration or activity of this enzyme. Okay? Why? Because it's present in in a high concentration in the liver. If you have alteration or increase of this enzyme in the blood, it means that you have a liver damage. What is the function actually? As I mentioned, aminotransferase are specific to each kind of amino acid. When we say alanine, it means that the donor of amine group is alanine. This is a alanine amino acid. Okay? Alanine donates its amine group to the alpha ketoglutarate. Again, all the time we have an alpha ketoglutarate as an acceptor of the free amine. Now, for example, for this reaction, alanine aminotransferase, the donor of the amine group is alanine. Alanine donates its amine group to the alpha ketoglutarate, okay? And alanine converts to the pyruvate, and alpha ketoglutarate converts to the glutamate. Clear? Or no? Repeat. I mentioned that the first step is the removal of the amine group. When alanine, this is the alanine amino acid, when alanine actually release its amine, now is a pyruvate. Pyruvate is acid, it's an alpha keto acid. Pyruvate is a carbon skeleton. Each amino acid has a specific carbon skeleton. For example, alanine is an amino acid. After removing amine, convert to the specific amine alpha keto acid. What is the name of this alpha keto acid? Pyruvate. Exactly. Okay? So each amino acid, after releasing its amine, convert to the specific alpha keto acid with a specific name. For example, for alanine, this is a pyruvate. And please pay attention to the role of glutamate. You know, glutamate, for example, this is the alanine, okay? Donates is actually, this is alpha ketoglutarate. This is the alanine. Alanine converts to the pyruvate and donates its amino group to the alpha ketoglutarate. Now it is a glutamate. Please pay attention to the role of glutamate. As you can see here, okay, glutamate is the lovely amino acid. Why? Because it carries all the amine. For example, glutamate carry all the amine group of other amino acids. It means that during the catabolism of each kind of amino acid, alpha ketoglutarate receive the amine and convert to the glutamate. Okay. Now, for example, I'm glutamate. Okay. This is the amine group. Okay. And I'm alpha ketoglutarate. After receiving amine from any kind of amino acid, now I convert to the amino acid glutamate. This glutamate collects all, for example, from alanine. For example, you donate your amine from alanine, from aspartate, from, for example, lysine. Okay, this alpha ketoglutarate collects the all amine from all kind of amino acid and convert to the glutamate. And this glutamate is a source of amine, okay? And in the next reaction, this glutamate became hydrolyzed and released its amine group, okay? And this amine group exerted into the urine as a urea, okay? It means that glutamate function actually and have a very pivotal and vital role because as a form of alpha glutarate, enter to the reaction, collect the amine, convert to the glutamate, and finally reach to the, for example, kidney and release this amine into the urine. Another aminotransferase is an aspartate aminotransferase. Please pay attention to the direction. Now, aspartate, for example, now it is aspartate. Okay? This is aspartic acid. Okay? At first, it should transaminate it and release its amine group and produce another kind of carbon skeleton. So when you have an aspartate, the remaining carbon skeleton is oxaloacetate. Okay? And again, alpha ketoglutarate participates participate in this reaction and don't actually receive the amine group of aspartate. Now it is glutamate. Glutamate actually can be hydrolyzed and liberate its free ammonia.